So I'm going to finish this section on transcription. And when I stopped, I was talking to you about how to stop these prokaryotes from continuing to make RNA off of the DNA. And I told you that oftentimes a repeated region was transcribed, and it formed a hairpin, which ultimately stopped this process. And then I also talked about this rho factor, where it translates along this piece of mRNA that's being created, and it's mRNA because we're in prokaryotes, and it catches up with your RNA polymerase and shuts it down. Now I want to talk a little bit about this kind of setup where, let's say you had a gene that you didn't want on in the first place, okay? And we might not need it because we're not exposed to a particular substance, or we haven't eaten it, whatever. Well, you can block RNA polymerase from actually binding or coming through in the first place. And this is a common technique, and I'll write this out, is I'll first put that you should understand that specific genes do not always need to be transcribed. And this is easier to understand in a single cell than it is for a more complex organism, but you know that there are certain genes that are on or off depending on what type of cell they are in eukaryotes. So specific genes, when they're not needed to be transcribed, you can use what are called repressor molecules or repressor proteins. Will bind to specific regions of the DNA and stop transcription. So if you can't bind your RNA polymerase or other molecules that might start it up, you're not going to make that particular structure, or you're not going to make that mRNA or hnRNA. So you can think of it as this is a gene that we want, a gene of interest that's down here, and we have this promoter region or some other area up here. If you have a protein that's bound here, so if this is some type of repressor, it's going to stop RNA. So if RNA polymerase is out here, or anything that might want to bind here, it gets blocked, and that gene doesn't get turned on. Well, you might not want that to happen forever, and so there are other molecules called inducers. And this binds to the repressor and causes it to dissociate from the DNA. So you can have this idea that there's something that's getting high enough concentration that's going to bind to this somehow, and this might be a little inducer molecule like a triangle here. Then it's going to bind here, and this would be the repressor protein, and it will cause that to change some type of conformation so it can no longer bind to this gene. And so the concept of repressors, you should understand that in inducers. And obviously there's lots of other ways that this can happen, but this is another sort of classic method for stopping it from even starting in the first place. And these are kind of prokaryote, like lower cell complicated systems. Eukaryotes, they even have like exonuclease that will start breaking down that piece of hRNA and catch up with your RNA polymerase and shut it down. I am going to give you a video of how this happens, and I'll just write down, I'll post it after this video, but when you think of eukaryote or eukaryotic transcription, it often requires more promoter regions and what we call transcription factors to turn on transcription. So it can get pretty complicated, and I have a video where it's awesome to watch this happen, but you can watch it and see as things get higher in concentration, they're more likely to bind and make this happen. So the last thing I want to cover before we start thinking about making proteins is, I've already told you that when you have a prokaryote, the RNA that's transcribed is mRNA, which means it's ready to go make a protein. So it directly codes for a protein. And it's a little different, yikes, for something that's a eukaryote. What happens is the RNA transcribed is heteronuclear RNA. So it needs to be what we call spliced. Okay, let me explain what's happening when you splice a piece of hRNA. Basically what happens is you have this piece of hnRNA, and let's say it's cut up into, and I'm going to put E1, I1, E2, I2, E3, I3. And the E's, they stand for exons. And we know that this is RNA that codes for a protein, or it will code for a protein. And then we have what are called introns, and that's what my eyes are. And this is RNA segments that does not code for a protein. So how do we get this into a situation where we can make a protein from it? And that's through what we call splicing. And splicing is a process of removing the introns and joining exons. And what's sort of interesting about this, and I mentioned this earlier, but this is done by those SNRPs. And that's not even fun to say, but a SNRP is, remember, the snRNA plus a protein structure. And the way it does this is it's complicated, but they have studied it and they know how this loops around. Your book does a little bit of an intro of how this takes place. But what's sort of brilliant about this is it conserves, like if you have a good scaffold for a protein in E1, you can make multiple proteins out of it by attaching different exons to it. And so you might see this piece of hnRNA be processed into a couple different things. You might see it connected if you took E1 and E2. That might be, now this is a piece of mRNA that codes for a single protein. Okay, and then you might have another segment where you connect E1 and E3, and that's a different piece of mRNA now that codes for, let's say this is protein A, and this would code for protein B, but it might share a region of the protein together, and you could even, have 3 different segments combined together, E1, E2, and E3, and that's another piece of mRNA that might code for protein C. So each one of those can be used to make a different component. And so I think I had, no, all I have here are different eukaryote structures and different binding structures and how these things connect with one another. But it's important to understand that these things can get pretty complex, but there's some redundancy and sort of efficiencies that are baked into these pieces of RNA. You can also understand that some of these non-coding regions, although they don't code for proteins, they're important for like the SNRPs to bind and to do this splicing appropriately, which I'm going to mention one last thing in this section. Years and years ago, if you read about the Human Genome Project, really was an effort to make sure that we had the genetic code of humans, and now we know we can get our DNA sequenced. Typically, they don't sequence it base per base, but they might look for regions or something. But we found out during that Human Genome Project that only 2% of DNA codes for mRNA that make proteins. And we know there's a lot of bases, so 2% is still a big number. They used to say that the rest of DNA, and this was a term that you might have heard before, and it still persists among a group of folks that haven't kept up with their genetic research, but the rest of the DNA they used to call what's called junk DNA. But it's not, right? We know that 80% of this junk DNA does a couple different things. It basically can interact with regulatory proteins. So things that are inducing or promoting, or you can think of this as promote, suppress, which is super important. If you get a mutation where you have a promoter, or you could underexpress. If you get a mutation where there's a suppressor, you can overexpress a piece of mRNA. So they can also be involved, so binding, they can also be involved with turning on or off genes. So you might say, well, how's that different than promoters or suppressors? Well, maybe there's not something binding. Maybe they're forming around a histone or another protein, or around themselves. They're kind of in a particular spot. But if there's a mutation there, we might not know exactly what binds to it. It might not bind to something, but a mutation in it can be involved or mess up a gene being turned on or off. Or they might not be either of these, but a mutation is associated with some form of disease. So we don't know why it is that way. That's another way of saying it's important. We just don't know why it's important. So DNA is complex, and the redundancy in it is probably, you might look at it and go, well, it's not efficient, but that might be true, but it might be important. So that's sort of a good lesson on life. So anyways, like I said, I am going to show you way down here on the bottom of this page, actually into resources, but I'll move it into this section, is this video on DNA transcription. So watch that, and then after I talk about translation, I'll put this video in there too.